gas from like fermentation,
Oh.
and then like things pop.
Okay. So, a couple of things I hear in there. Fermentation, spoilage. What do we mean by spoilage? So when something spoils, what's the cause of that? Typically bacteria, maybe if you're a microorganism of some sort, right? If you have bread and you let it sit out too long, what happens to bread? It molds, right? That's a fungus, obviously, but we have microorganism being involved. Now, anybody into home canning? Ask every semester, and usually it's one person, if anybody. Okay. Anybody know somebody who does home canning? Grandmas, grandpas. Okay. How is canning done? So you take all your veggies, you throw, or whatever fruits. You just eat it. Okay, that's fine. Has anybody seen the process done before?
Yeah, you put it in boiling water, and then you gotta wait for the thing to pop.
Yep.
And then you take it out and then seal it.
Okay. Yeah, there's a couple of steps in there, and the first one, let's hit on heat for a minute. So we're talking about ways to inhibit growth. Heat is one common mechanism, and in fact, in the homework assignment, there were one of the mechanisms in there, pasteurization, uses heat as a way to control microbial growth. Heat's going to kill or inhibit the growth of what kinds of microorganisms? So which ones don't do well at high temperatures? Psychotropes won't, so we'll kill off psychotropes. Psychrophiles. What ones are we most often, we're concerned oftentimes with mesophiles as well. So the heat step there, the goal is to inhibit the growth of microorganisms, hopefully kill them with that high temperature. Canning like this is done in a similar method. We heat up, maybe flash heat the pears for a short period of time, and can them in an anaerobic environment. That lack of oxygen is going to inhibit the growth of what type of microorganisms? Which ones need oxygen to live? Obligate aerobes. Facultative don't necessarily need it, right? They can grow with it, but do they need it to live? They can live without it. There's two main ones that require oxygen to live. Obligate aerobes and our microaerophiles. So in a process like canning here, or if you're doing it, if you're doing home canning, that heat helps to chase out the extra oxygen from the cans. You seal it shut, and now there's no oxygen present. So you're inhibiting the growth of some of those spoiling bacteria. But what can still grow? What could still grow in that anaerobic environment? Facultative could. What else? Obligate anaerobes. One more. Yeah, our aerotolerant would be able to as well. Okay. So in this can here, Mancini, you pointed out fermentation is occurring, and that fits. Lack of oxygen would lead to some sort of fermentation event. That's producing a gas. I'm sure if we tested the pH in this can as well, it probably would be reduced to some extent from the fermentation process. But given those two, given the lack of oxygen and given that heat step, we're still finding that there are signs of microbial life. What kind of microbe is able to do this? And it's a pathogen. So it's a mesophile as well. So what's gone wrong? Why is a mesophile, even after heat treatment, able to What are some characteristics of this organism? Or this genus of organisms? Okay, it's an obligate anaerobe. That would explain why it's capable of growing in the absence of oxygen. So obviously that removal of oxygen isn't a very good method to inhibit its growth. What are other characteristics? Gram-positive? I think if I'm not mistaken, it's a bacillus. Other characteristics? Where do we find them? Where is Clostridium oftentimes found? In the intestines if we're talking about C. diff, but it doesn't just get there unless you're talking about hospital setting. Where's the natural environment for Clostridium? Water can be a possibility. If you step on a rusty nail, what are you told to do? Get a tetanus shot. Why? Is it the rusty nail we need to be worried about? No. It's the microorganism that's found in the soil with it. Oftentimes, Clostridium are found within the soil. Okay? And actually, some of the worst Clostridium outbreaks are ones that occur with potatoes.
What? Isn't it found in poop?
Well, it is, yes.
And then don't we use manure?
Yes, but I'm going to put a question mark here because yes, you're right. We will find it in human fecal sample. I don't know if we'll find it in cow fecal sample. I just don't know. But I will put that there with a question mark because it's a possibility. All right. Clostridium, one of these features here allows it to survive that heat treatment. Which? Sporulation. What is a spore? What's that? Okay. To protect it. So it's a mechanism of protection, right? What else? What types of organisms can form spores? We've defined this once already. There's only one type of microorganism that can form a spore. Gram-positive and their bacillus. The two major types that form spores are the genus Clostridium, and then the genus Bacillus is the other one that form the majority of spores. So with Clostridium, we have three types that we oftentimes talk about in terms of causing disease. Clostridium tetanus, Clostridium tetani, which causes tetanus, Clostridium botulinum, which causes botulism, and then Clostridium difficile. Spores are incredibly challenging to get rid of using physical control methods. They're also really challenging to get rid of using chemical control mechanisms that we'll talk about on Monday. Heat can work, but oftentimes it has to be heat plus pressure. Boiling water, it takes 2 hours in just boiling water to kill a spore of Clostridium. But if you apply pressure to it, so that the temperature of that water before it boils is up to maybe 120 degrees Celsius, now you can cut down that time to 5 minutes. So in terms of home canning, it's often suggested that we use pressure cookers for the canning process rather than just boiling water itself. Okay. Spores are also incredibly resistant to things like UV light. And Clostridium, whenever oxygen is present, what you will find is it begins the process of sporulation. So sporulation is a process in which a microorganism, like Clostridium, instead of dividing by binary fission into two daughter cells, will begin to form inside the mother cell an endospore. And these endospores have very thick coats. These thick coats help to protect against chemicals and different environmental effects. The DNA is tightly bound up to prevent damage to it. And then there are some additional molecules that are produced to help this spore to survive cases like desiccation. What is desiccation? Yeah, removal of water, exactly. So spores are incredibly resistant to these environmental factors. One of the reasons why C. diff is so challenging in hospital settings right now to get rid of. What are some possible reasons? The simple answer is it sporulates. And the spore, contact with Lysol on a towel for a short period of time, not enough to kill a spore. We have to use much more harsh chemicals to kill a spore than we do a vegetative cell, one that's actively growing. What does this image have in common with this image? This is from a commercial for migraine treatment. Anybody know what the newest trend is for migraine treatment? Botox. What is Botox? It's botulism toxin. Okay. So I don't know, I can't remember where I was going with this. Probably just to point out that the same organism that causes cans to explode can also be used for facelifts or for migraine medication. Okay. The difference between the two is the toxin, so botulism toxin botulism toxin causes what we call flaccid paralysis, whereas tetanus causes like a rigid paralysis. We'll talk about that later on in the semester, but I figured I'd bring this up anyway. All right. Don't lie down, stand up. Okay. Where did I want to go with this? Let's talk about a couple of these control methods. When we're talking about physical controls of microbial growth, I had you guys look at 4 specific examples, but what are the themes of those 4 examples? So pasteurization. What is the theme behind pasteurization? What are we actually using to kill the microorganism? Heat. So changes in temperature can have an effect on a microorganism. Sorry, this disconnected. There we go. Desiccation. We've already addressed, right? Desiccation, removal of water would be another one. And as water is removed, what does that do to concentrations of salts? Increases salt concentration. So removal of water can be used as a good way to inhibit the growth of what types of microorganisms? Would a halophile die as salt concentration increases? Why not?
Because it can live in high concentrations.
Good, good. So we'd want something that will die or that grows only at low concentrations of salt. Non-halophile would be a great example there. What are examples of how we use desiccation? We use it all the time in the food industry. What are examples of it? Where we have either low concentrations of water or high concentrations of solutes so that the water is bound up and not accessible to bacteria. What are some examples? Grace.
Raisins and beef jerky.
Raisins, beef jerky. What else? Those are 2 good ones. Any dried fruit would work, yeah? Even things that we don't necessarily think of as dry use the same principle. Fruit preserves or jelly. The water is bound up in the gelatin that's used to make jelly, or the water is bound up with the high concentrations of sugar, and that can inhibit microbial growth. What's the best friend to jelly? Peanut butter. Peanut butter is another example where we see desiccation as having an effect. So desiccation and raising the concentration of solutes have a lot of correlation with each other. And both of those can be used to inhibit the growth of non-halophiles. Okay. So desiccation, we've got temperature with pasteurization. I'm missing a couple. How do we use light or radiation? What are examples of radiation and how they're used to inhibit microbial growth? UV would be one. Microwave can be used, but in what way? What happens when you stick something in the microwave? It's heated. I think it can. But oftentimes, most often what we talk about in here, there's two main mechanisms that we use. One is UV. What's the other one? Well, let's just start with UV. UV's good for surfaces. UV does a great job of sterilizing surfaces because, well, what does UV do to a cell? What effect does it have? It obviously blocks the growth, but why does it block growth? Messes with the DNA. Okay. Same reason why we in the U.S. argue that people should not go tanning. It ruins the DNA. Okay. The other form of radiation would be ionizing radiation. So X-rays, gamma rays. These are really good for penetrating within a substance. So there are some that argue that if we want to stop the number of outbreaks that occur in leafy greens like lettuce and spinach, where we see time and time again outbreaks of salmonella illness, for example, we should actually be using ionizing radiation to sterilize those vegetables. Of course, the freak out is going to be, wait a second, doesn't that do long-term damage? And the answer is no. The damage is only there as long as that pressure or that ionizing radiation is being applied. The effect is on DNA, causing either thymine dimers or double-stranded breaks in the DNA that can't be repaired by the microorganism. So it inhibits growth in that mechanism. We are missing one more. What is the last one? Filtration can be, that's actually a good one. Let's talk about filtration for a minute. What is filtration? Removal of microbes, how? Or based on what? Yeah, based on, another way to say it is it's inhibiting microbes from moving from through with the solution. Oftentimes this is used with liquids or gases as well, but it's excluding them based on their size. So a bacterium typically is about a micrometer in diameter. If we create filters that have pore sizes smaller than that, we can inhibit their movement with the liquid. Where do we see filters being used in hospital settings? And don't think liquids. I'm going to put up 4 letters real quick after I do this, because I just noticed I can do this. Okay, sorry. What's HEPA or HEPA? Nobody's heard of these?
Like an air filter.
It is an air filter. Why would that be important? Why would we want to filter air in a hospital setting? Airborne disease, absolutely. Somebody coughs, you don't want that microbe being able to spread throughout the whole hospital, and so by circulating purified air, we can reduce the potential for spread of airborne disease. And this is used all the time in hospital settings. So this would be one potential example. What? Throughout the whole hospital, pressurized rooms are a little bit different, Michelle. So those are actually quite common too. Or another example of how you can use filtration. So those pressurized rooms, if I'm remembering correctly, pump the air out, and the microbes get captured in the filters as that air is being pumped out, so that it can then be recirculated through again. So you do see them being used in that process as well. Actually, at UW, whenever you have a research laboratory that's using microorganisms like a BSL-2, so something that causes disease, they will have those pressurized rooms to help so that the microbes aren't spreading in the room itself, but they're always kind of being collected into the filter, and then that air is recirculated. Yeah, that's a great question. One last one that that we haven't really talked about is affecting oxygen contents. So, for example, we just said we said a little bit earlier in the canning process that can be used as a way to inhibit the growth of obligate aerobes and can be used to inhibit the growth of microaerophiles. If you are like my family and have a lot of deli meat in the house, oftentimes that deli meat comes in vacuum sealed packages. And that vacuum sealing, again, the benefit of having vacuum seals is to inhibit the growth of aerobic microorganisms, ones that require oxygen to live. We also see many other types of control outside of this. What type of control might we find in, say, yogurt? What are the characteristics of yogurt?
It's already fermented.
What do you mean by that? Let's help out Emma with this one. You're right. It's already fermented. So, what kind of fermentation really is a different way to ask this? What kind of organisms do we find, or what kind of bacteria do we find often in a vat of yogurt? I know you guys know this answer. I'm just going to draw a vat of yogurt in the meantime. That's a big one. Lactobacillus. What do lactobacilli produce in their fermentation process? I hear some people saying
Lactic acid.
Thank you.
You're welcome.
Lactic acid. Lactic acid has what kind of pH? Acidic, less than 7, probably around 4 or so by the time that the fermentation has been completed. So we can use acids to inhibit the growth of neutrophils or achillophiles as well. So using even a microorganism's natural metabolism can be a way that we can inhibit growth. And we see this time and time again too. Anybody like pickles? Love pickles. That was the correct answer, Nancini, is you love pickles. Anybody like sauerkraut? We hate sauerkraut, okay? Maybe you like it, I despise it. But both of those use acids, either added to the pickles directly, or added to the cucumbers, or natural fermentation from lactic acid bacteria to produce the acids that inhibit the growth of microorganisms. Let's go over a couple of specific examples. Pasteurization versus something like autoclaving. Autoclaving, we have one of these. We use it to sterilize all the media in the lab. How does that differ from pasteurization? And maybe I'll give real quick what the conditions of a autoclave are. 20 minutes, 120 degrees Celsius, pressurized. How does that differ from pasteurization? Well, there's bacteria involved in all of these, technically, right? But what do you mean by that, Emma?
Like, do they use the bacteria?
No, they don't. Thanks for clarifying.
Is it that the heat kills a lot of bacteria?
In which of the two?
The first one you talked
Pasteurization or autoclaving?
about. Autoclaving.
Okay, yes, but pasteurization also kills a lot of bacteria. If we were to look at just raw numbers, both of them are really good at killing bacteria. So it's more specific than that. Take a look at how pasteurization is performed.
Is it
Taylor.
like flashes of heat, like over and over again for bacteria, like really high heats?
Okay, so what does that do then?
Kills the bacteria that aren't used to like the flashes of heat, like
Okay.
aren't able to adapt fast enough.
Good. So pasteurization, here's another way to think of it too. You buy a carton of milk at the store. Oftentimes you will see on there that it's listed as being pasteurized or ultra pasteurized. Okay, and there's a slight difference there. We'll talk about that in a minute. Does milk spoil?
Yes.
Yes, it does. If I have a container of LB broth sitting on my lab bench, leave it unopened for 3-4 years, and it's been autoclaved, it will not spoil. It will not support microbial growth. What does that tell you? So if milk is still able to spoil, what does that tell you about what's in there after the process of pasteurization? Not everything is killed. There's still bacteria in there. Whereas with autoclaving, the goal is to sterilize, to remove all microbial life. Pasteurization is a method of what we call disinfection. The goal is to kill pathogens, to kill the majority of microbial life, but we may not kill all of the microbes. So in pasteurization, the two techniques that we see most frequently nowadays: heat at 72 degrees Celsius for 15 seconds, or ultra UHT, ultra high temperature pasteurization, UHT pasteurization. Main thing to keep in mind here, one, you need to know the characteristics of the organism that you're trying to kill. You need to be able to know, is it a mesophile, is it a psychotroph, is it a thermophile, and that can help you to decide what is the best condition, or what might be one of a few good conditions to inhibit the growth of the microbe. The second point, and we'll hit on this some more when we talk about chemical controls of growth on Monday. Two things are really important here: time and magnitude of the effect. And what I mean by that is, we just said with pasteurization, 72 degrees Celsius for 15 seconds, or 140 for 3 seconds. As temperature goes up, what happens to that amount of time that's needed for the exposure? Decreases. And we find this time and time again. So those two features, the temperature is 72 versus 140 is the degree or the magnitude of that effect. that will affect how much time is needed to kill the microorganism. And so keep that in mind as we start to move into discussing chemical controls of growth on Monday. Okay? Monday's the mini lecture for Monday is a little bit different. There's about 9 slides. I talk for 4 of them. The rest of them is informational. Take notes, look at the book, and we'll use those as our starting point on Monday. All right. As you're heading out, if you want, I have quizzes and your tables from yesterday. So feel free to grab them if you want to. Page.
Do we have to inoculate for tomorrow?
No.
Or not tomorrow?
No, you don't have to do anything for that.
For Tuesday? Then?
No, we're just going to use the cultures that you guys have. There you go. Oh, she's not here. Rosie, did Rosie? Damn it. Okay. Well, I guess I'll just give these back later then, unless people want them. That's cool. Allie, I do have your quiz. There we go. There you go. All right, Lindsay. Go. Oh, yeah, go ahead, please. Sorry about that.
See you next week.
There
Thank
you
you.
go.
So, thank you.
Yeah.
Should we change any of this? Should we add more?